Welcome to the second part of staining techniques. Last video we have studied about the various methods of staining. We have classified them and we have studied about simple staining and differential staining. In differential staining we have studied about identification part, that is gram staining and acid fast staining. Today's class will be studying about the visualization differential staining technique, which include endospore staining, okay, and after that we'll be studying flagellar staining, nuclear staining, and the capsule staining, okay. Beginning with endospore staining. What does endospore mean? Endospore is outer covering, which are like tiny spores. Okay, they have very thick wall, and these endospores hardly absorb any dye, and if they absorb, they easily doesn't let it go. So we'll see what endospore staining is. Okay, and spores are thick wall structures and resistant to physical and chemical agents. Doesn't matter what you add, nothing will happen. Suppose if a stone is there, if you try to hit a stone with another stone, does it happen anything? It won't happen. Doesn't happen anything. It won't even affect it, right? Similarly, these spores are so strong that they won't be affected either by physical or chemical agents, and they survive for longer periods. That is, we call them as dormant state. Okay, and these are also heat resistant. Why are they heat resistant? Because the spores are made up of calcium. Okay, calcium dipofolinic acid. These are when this is deposited in high concentration, forms a very thick wall. As a result, it is very strong and doesn't get affected by any other agents. Okay, and because of this reason, we need special techniques in order to identify them or visualize them. We'll see that. An aqueous primary stain, that is malachite green, is applied and steamed. The reason for adding a malachite green and giving a steam is to enhance spore pore penetration. Because of steaming, this steaming helps. Basically, if we take something, when we heat it, it gets loosened, right? So when we heat, because of that steam, that heat, the particles slightly move away. As a result, there will be a small space, and that will be helpful to penetrate the color into the spore. Okay, and once stained, the endospore does not readily decolorize because endospore is very strong. When it absorbs the dye or any color, it easily doesn't let it go. Even with the application of decolorizer, and they appear, but the cytoplasm of the cell takes the color and of saffron appears there. So what it means is, if we are adding some color, that will be absorbed by the endospore because of heat, and if the same color is taken up by the cytoplasm, it will be same in color. So there should be some different color. So when we add some other color, the cytoplasm present will be absorbing that color. As a result, the endospore has a different color and we will be able to identify it. So what is the next point? A modified Ziehl-Neelsen stain. Okay, a modified Ziehl-Neelsen stain. The scientist name is Ziehl-Neelsen. He has prepared that stain that is used along with 0.25% of sulfuric acid as a decolorizer. When this is applied, a red color uh, spore is formed in blue stained back. Okay, uh, what it means is, if a bacteria is blue stained and the endospore is absorbing light color, it will be showing as a blue color. Okay, we'll see the procedure now. First, application of malachite grade. We'll take a smear. What is smear? Heat fixed smear. What is it? Slide onto the slide. We add the microorganism. Okay, loop of microorganism solution or suspension and heat it. As a result, bacteria retain there. That is called a smear. To the smear, we add malachite green. Okay. After that, we heat it. Okay. This heat makes the color to be absorbed properly. As a result, the bacteria will absorb that green color, the malachite green. It will be absorbing the green color, and when we rinse it with water, everything goes away, and only the bacteria which absorb color will be seen. After that, when we apply saffron into it, after that, when we apply saffronin, that saffronin will be absorbed by the bacterial cell, whereas the spore will be retaining the green color, because spore doesn't let the color easily go. Once it has absorbed some color, it won't let any other color to form on that. Okay, we have something, or we have if we have something very important, we won't let others to take it. Similarly, sports, when they imparted their certain color, it won't let go away, and it won't let other colors to bind to it. Okay. Uh, you can see the procedure on to the right. Place a slide over a beaker of boiling water with bacterial film uppermost. That which means bacteria, if it is a slide on the upper side, the bacterial culture will be there. Okay. This one will be putting onto the beaker of boiling water. When large droplets condense on the underside of the slide, which means below the slide, if the droplets, the vapor coming forms here and forms a watery like, then flood it with 5% of malachite green. Malachite green is the dye being used. After adding that, leave it for 1 minute and rinse with water. Okay. And okay, after that, rinse with cold water, then treat with 0.5% of safranin or 0.5% of basic fusion for 30 seconds. What is the reason of for adding safranin or leucine color, basic leucine? It is to impart color to the bacteria, the pinkish color to the bacteria. Therefore, after that, we will be able to identify which is the spore part and which is the bacterial cell. And after that, we should wash it and dry it and observe under microscope. So how does it look? Spores look green in color. Okay, you can see here the green color part, that is the spore, and the vegetative bacilli are red, which are growing bacteria. Okay, and lipid granules are unstained. The lipid granules will be able to see just like a droplet, just water, colorless. Okay, that will be lipid granules. Okay, next is capsule staining. What exactly is capsule? You might have heard about it. The bacterial cell will be having capsule, capsid around it. What is it? It can be either a polypeptide, which is a protein, or it can be a carbohydrate. Okay, that means a sugar, or it can be a polysaccharide. Sugar means polysaccharide, and it can be glycoprotein. So basically, capsules are those which contain uh, polysaccharides, glycoprotein, and polypeptide. Either of them, okay. And these capsule materials are water soluble. Because of that nature, it will be difficult to identify under microscope because we'll be washing with water and we'll be trying to impart color. If we impart color, even if it binds when we wash with water, it will be vanished away completely. As a result, we won't be able to identify them. So some special technique should be needed for that. So in this, for this capsule staining, heat should also not be supplied. If we heat, since it is a sugar molecule, it contains polysaccharides or proteins. As you know, proteins are very heat sensitive. They easily get denatured, right? Even polysaccharides, the sugar molecules are very sensitive to heat. They just get degraded. As a result, we should prevent heating it. That is, when we prepare the smear, we should not heat. We need to air dry it in order to retain the bacteria. Okay, and non-ionic dyes, the bacterial cell, the capsule is non-ionic, therefore dyes do not bind to it. So when dyes are not binding to it, what do we need? How do we impart color? In order to impart color, the substance should have a charged, okay, it should be either positive or negative charge. So how can we impart that? Suppose I take a magnet and take an iron nail. When I put magnet close to the iron nail also behaves like a magnet, right? Similarly, to the capsule, which are non-ionic, it doesn't have any charges on it, either plus or minus. When I get any positive or negative charge around the capsule, that capsule also start behaving like a polar one. Okay, so for that reason, acidic or basic dyes are used to stain the background cell wall. So let us see the procedure. There are basically two methods: negative staining method, okay, and positive staining method. We'll see what they are. Negative staining methods contrast a translucent, darker colored background with stained cells but an unstained capsule. What it means is bacterial cell absorbs the color. Okay, suppose if I add crystal violet, it absorbs blue color, and the cytoplasm has certain color. Okay, it will be a lighter one. The bacterial cell has dark color, the cytoplasm has lighter color, but the capsule around the bacteria will have no color. Okay, by this we'll be able to find the thickness of the capsule. Okay, and we can be able to see what type of capsule it is. Okay, and the basic dyes used in this are India ink, nigrosin, and congorate. Whereas positive capsule stain requires a mordant that precipitates the capsule. What is mordant? That means we need to put a beaker bottom and heat it so that the capsule gets precipitated. By counterstaining with dyes like crystal violet, methylene blue, or carbon fusion, bacterial cell wall takes up the dye. Okay, when we add crystal violet, bacterial cell wall takes up the dye, and capsule appear colorless as similar to negative staining, and the stain cells dark against dark background. So basically, in negative staining method, the background was lighter and the cells were having darker color, and the capsule were colorless. Whereas in positive staining method, the background is darker, the cells are having different color, and the capsules have no color. Let us see the procedure now. First, inoculate the loop. Inoculation means addition of loop of culture media onto the glass slide. Okay, prepare a thin bacterial smear. Okay, then air dry it as capsule is heat sensitive, contains polysaccharides or protein. It will be degraded, so we need to air dry it. After that, apply 1% of crystal violet. You can see here heavy smear prepared and air dried. Okay, after that, with the help of a dropper, we flooded with crystal violet. What is the percentage? 1% of crystal violet. After that, copper sulfate solution is added. This copper sulfate solution should be added in excess so that the excess crystal violet gets binded to the copper sulfate and gets removed. Okay, so wash the crystal violet with 20% of copper sulfate, and then take another slide or a cover slip or a paper which will be able to absorb the extra solution present. Okay, not the slide or the cover slip, just take a paper, blot dry. What does it mean? We need to take a paper, put slowly so that the extra solution present gets absorbed by the paper and retains the bacteria on to the slide, and then examine under the microscope. How does it look? In this way, you can see the lighter background. Okay, the lighter background, the darker cells. These are the darker cells which because of crystal violet, and the colorless surrounding the bacterial cell is the capsule. Okay, so this is the background. This is the white color is the capsule, and the bluish color are the bacteria. This was capsule, and other techniques include flagella and nuclear technique. So we'll see what flagella techniques are. First, grow bacteria for 16 to 24 hours on a non-inhibitory medium. Example, blood agar medium. It is a non-inhibitory medium. Okay, or a tryptic soya agar. This is a different type of agar, Newton medium used. Okay, first bacteria should be grown for 16 to 24 hours in that specific Newton medium. Okay, touch a loopful of water onto the edge of a colony and let motile bacteria swim on into it. What it means is, take a slide and put some solution of bacteria, okay, and uh, add a drop of water to the end so that the bacteria that has flagella can swim into the water and gets away. Okay, bacteria having flagella will be swimming into the water and get away. Then transfer a loopful into a loopful of water onto the slide to get faintly turbid. Okay. Again, after doing that, add one more drop of water onto the bacterial smear so that it forms a turbid. When the water is contaminated, filled with microorganisms, it shows turbidity, right? So when water is added into it, it shows turbid nature. Okay. Then the bacterial suspension is thus prepared with the minimum of agitation. We just need to shake like this so that it will be completely mixed with the water, which would discharge as a flagella. During this agitation, during this movement, the flagella gets separated. Suppose we have some cereals with us and we want to separate stones from them, we just shake in this. Usually this happens in villages. They keep shaking like this so that the stones get separated from the actual cereals, right? Similarly, when we agitate the glass slide having a bacteria into that, this process leads the removal of flagella from the body and gets separated. Okay. After 5 to 10 minutes, when many bacteria have attached to the surface of the slide and cover slip, apply 2 drops of reused stain to the edge of the cover slip and leave the stain to diffuse into the film. What it means is, after we get a turbid solution onto the glass slide, we need to agitate it, then put a cover slip, okay, cover slip onto it, and then add, beside the cover slip, add Ruiz stain, okay. As a result, that stain get absorbed, not absorbed, it diffuses into the bacterial solution which is below the cover slip, okay. That diffusion helps in absorbing the color. Examine with the microscope after standing for 15 to 10, 5 to 15 minutes. So after putting the cover slip and adding a Ruiz stain beside, wait for 15 to 5 to 15 minutes, 5 or 10 or 15 minutes, and then observe under microscope. By this, we will be able to see the number of flagella present. How can we see? Because of that Ruiz stain present. Okay, this Ruiz stain helps in identifying flagella. If it is one flagella, two, three, four, it means we can find the type of bacteria having different types of flagella or number of flagella. Based on that, we can differentiate into locotrichus, peritrichus. Okay, in that. So you can see the image on the right side. So this 29 there has one flagella on both the sides. Okay, the second one is the same. It has uniflagella monotrichus. Okay, and it has on lateral sides many, and here one, and you can see the entire body has many, right? Here and this one. So this type of this method helps in identifying the various bacteria having different flagella on it. The next method is nuclear staining. What is nuclear stain? The term nuclear itself means the nuclear membrane or nucleus having the DNA or RNA. The nuclear material or genetic material are DNA and RNA. So we'll be staining that nuclear material. And how do we do that? With the help of Giem's method or Giemsa's method. Okay. The basic principle, let us see what it is. Giemsa's stain is prepared by mixing two stains that are methylene blue and eosin. So we are using two stains, which is one is acidic, one is basic. That means a neutral stain is prepared using two polar stains, that is methylene blue and eosin. Both are mixed to form a neutral stain. Okay, so the resulting stain has properties of both the dyes. So cationic and anionic both properties will be present in this neutral stain. The chromatid of a cell is highly acidic in nature. So when it reacts with Giem's stain, it gives a reddish purple color to DNA. As discussed, if it is a negatively charged particle, it binds to positive. If it is a positive charged particle, it binds to negative. So chromatid is highly acidic, which means it has a negative charge on that. When it has negative charge, it will bind to a positive charge, isn't it? When it binds to the positive charge, that color will be imparted. So because of imparting that color, due to Giem's stain, the DNA or RNA will show color. The RNA of a cell is removed by acid hydrolysis step. In this step, the smear is treated with one normal HCl. Okay. After we apply this dye, we need to proceed with hydrolysis step. How do we do for hydrolysis? By putting the slide in one normal HCl. Okay. One normal HCl solution. When we put the slide, the acid, because of acid hydrolysis, all RNA will be removed. And then we need to and during this process we need to provide heat, that is by boiling in a water bath at 60 degrees centigrade. Whereas DNA material has triple bonds in between some base pairs, so DNA molecule doesn't get hydrolyzed, and hence only a DNA molecule stain and cytoplasm remain colorless. What it means is RNA, it undergoes hydrolysis. Okay, as a result, it won't be there. Whereas the DNA, which has double and triple bonds between the nitrogen bases, because of presence of that triple bond, hydrolysis doesn't occur. As a result, DNA will be absorbing the color and imparting its color, thereby visualizing it. And whereas the cytoplasm will be uncolored. So let us see the procedure. First, prepare smear, air dry, fix by using Boyne's fixative for 15 minutes. Buoyant fixative is a different procedure for heat fixing of the bacterial cells. Okay. Then place the slide in one normal HCl solution in water bath for 5 minutes at 60 degrees centigrade. Then rinse slide with water. Flood with Giemsa's stain. Okay. This is the main stain which is used for the DNA of genetic material to absorb the color. After adding the gem stain, GMSA stain, we need to wait for 3 to 5 minutes and wash with water, observe under microscope. How does the microscopic picture look? You can see here the bluish color is the genetic material. It can be DNA, okay? And the cytoplasm appear colorless and the nuclear material appear purple in color. In this, you can see the purple color, which is genetic material, and these are other organelles. And in this case, you can see the bluish color, whatever is there, that is the genetic material, and the rest of the part is cytoplasm. Okay, and this was with today's class. So we have studied about staining methods, simple staining, gram staining, simple staining, then differential staining. In differential staining, identification, visualization. In identification, gram positive, gram staining, and acid fast staining. Okay. In gram staining, we will be able to find if it is gram positive or gram negative bacteria. Whereas in acid fast staining, we will be able to differentiate mycobacterium species. Okay, acid fast bacteria and non-acid fast bacteria. Whereas in visualization, we studied endospore and the capsules, endospore staining, capsule staining, then flagella staining, and nuclear staining. In capsules staining, we were able to see colorless capsule around a colored bacteria and a lighter background. Okay. Whereas in a flagella staining, we were able to find how many flagella are present. Okay, with the help of Ruiz stain method, Ruiz stain. Okay, and in what was the next one? The next one was flagella, endospore staining. Okay, we have also seen endospore staining, and then we have studied about the nuclear staining, where we were able to see the nuclear material being colored in purple color against light cytoplasmic background. Okay, this was with methods of staining and various techniques. In the next video, we'll be studying about sterilization. What are different methods of sterilization, physical, chemical, and mechanical, and what are the merits, demerits, and all. Okay. So thank you for watching the video.